I defended my PhD a few weeks ago. Yeah, it still hasn't sunk in at all. To be totally honest, I was so stressed, so the day of my defense is a bit of a blur, but here's generally how it went down. So after sending my dissertation to my committee, all 170 pages of it that describes the research I did while I was in graduate school, I then had to give a public talk to present my work, followed by a private meeting with my committee to discuss the dissertation. So the talk and the meeting is the defense. Spoiler alert, I passed and am now Dr. Edwards. What? Anyway, I thought I'd share the talk with you all in case you'd like to know what me and the flowers have been up to at Harvard for the last 7 years. Hope you enjoy.
It is my great pleasure to introduce Miss Molly Edwards to give her PhD defense in the Department of Organismic and Evolutionary Biology. So as some of you will know, back in August when I introduced what Molly likes to refer to as her lab twin, Minya, for her defense, I gave a little preview of today. And I said that I had picked this, I had this song picked out from Minya, which was a song called, I think it was called Invincible. And I also had a song picked out for Molly. So she has been waiting since then to find out what her song is. So now I get to share it with all of you. And of course, it's doing that thing where I have to do this. So let's see if I can display.
Just kidding. The song is Baby I'm a Star by Prince. So in order to avoid copyright issues, I'm muting Elena's audio and replacing it with a robot voice and some stand-in music, but I'll bring her real audio back once the song is over.
Okay. So this is Molly's song. And so I hope that I can convince all of you, if you can hear me over the song, I hope that I can convince all of you that Molly is a star. So Molly did her undergraduate at Cornell, where she worked in Mike Scanlon's lab. She worked on meristems. She did an internship at NYBG and was a tech at NYU. When she came to Harvard, she decided to work on petal spurs, which is what she's going to talk about today, so I'm not gonna steal her thunder. But she all along has been running a science communication website on YouTube called Science in Real Life. Just one of the reasons why she's a star. She's also a lab star. She's had fantastic success with her research. Had a first author paper in Evolution last year. She's won awards for her work on Science IRL. She's won awards for speaking at conferences. She's won awards for her teaching. She is just overall a fantastic graduate student. She is also a fantastic friend and a fantastic member of our community. So everyone loves her and thinks that she is a star. She also is deeply dedicated to plant biology, as you can see from the plants behind her. She is a little bit obsessed with plants, and I think that will come through in her talk today. She's a little bit shy, surprisingly. You know, if I had to say, what are some surprising things about Molly? I'd say she's a little bit shy. Also, Minya tells me I would be remiss in not mentioning that she has a very bad habit of leaving too many windows open on her desktop. This is the worst thing we could find to say about Molly, that she habitually leaves too many windows open on her desktop. So I know we're all really looking forward to her talk, so I won't take up too much more of our time. But I hope I have convinced all of you that Molly is our star. And we are also looking forward to this event today. So the bad news is that I have to turn off the music now.
All right, I'm gonna turn off the music and I'm gonna hand things over to Molly. So after seeing her talk, you will also be convinced that she is a star. So go ahead, Molly.
Oh my gosh, thanks so much, Elena. Thank you. I'm very overwhelmed. Okay, does that look okay to everyone? Amazing.
Yes.
Well, team, thanks so much for being here today. I am going to set the tone of this talk by sharing what I think is one of the most pure and truthful botanical song lyrics ever written, which is, it just resonates so much with me. I've been learning lots of things from the flowers my whole life. They've been an endless source of joy and fascination for me, especially in the last 10 years that I've been studying their biology. And as you get to know the flowers really well, you start to see that each one will teach us something unique. So we're going to learn very different things from this flower than from this flower. Most of what we know about flower development, we learned from this flower, which is Arabidopsis. And it's kind of like the plant equivalent of the lab rat. So it's the model system for plant research. We've learned so much from it about how plants work. It continues to teach us so much. But it does make flowers with very simple floral organs, like these teeny tiny flat petals that don't really represent the magnificent complexity and diversity of floral morphologies, floral forms that we see in nature. And on top of that, Arabidopsis doesn't rely on animal pollinators to reproduce, but we know that pollinators have been this major driving force in flowering plant evolution. So if we want to learn a lot of things from the flowers about complex morphology and pollinator-driven evolution, things that will help us understand floral biodiversity, we need to learn them from a different type of flower. And that's why we in the Kramer lab study the genus Aquilegia, which is also known as the columbines. And one of the main reasons is that they make these beautifully complex petals. So let's take a closer look. Imagine I've plucked one off and laid it on its back. You can see that they form these elongated three-dimensional spurs. There's a nectary at their tip that secretes nectar into the spur as a reward for pollinators. And then extending upwards from the attachment point is a laminar, a flat portion of the petal that we call the blade. Extremely early in development, when these petals are still wrapped up in a flower bud, they're very flat, resembling something like you might see on an Arabidopsis flower. But a little later on, an outpocketing starts to form that will eventually become the spur. Imagine one more time now, we've taken these petals and turned them on their side and then made a really thin slice through them. That's the view that you're seeing in these microscope slides that have been through an in situ hybridization experiment to show histone 4 expression. This is just a cell division marker. So basically anywhere you're seeing purple dots, you're seeing cells dividing. And these show that cell divisions are happening diffusely throughout the entire petal at this earliest stage, but they soon cease in the periphery and become concentrated at the site of the nascent spur. So we've dubbed this phase 1 of spur development, which is characterized by this concentration of divisions to form this little outpocketing. Cell divisions cease in the spur by the time it's only a few millimeters long, and the remaining 90% of spur length is reached during phase 2 of development, and it's achieved solely through highly anisotropic, highly directional cell elongation. So not only do their petals undergo this unique developmental process to generate the spur, there are roughly 70 species in Aquilegia exhibit stunning diversity in spur length and shape, as well as a lot of other floral traits, depending on their pollinators. So the genus is widespread throughout the Northern Hemisphere, and it originated in Eastern Asia, where almost all species are bee pollinated. But then things got really interesting when the genus migrated to North America around 3 million years ago and encountered new pollinators. So this is a phylogeny, or an evolutionary tree, of the North American columbine species. And these colors represent pollination syndromes, which are these suites of floral traits that attract and reward specific pollinators. And they've been analyzed in the context of this phylogeny to determine how they evolved. So the bee pollination syndrome is characterized by blue-purple flower colors. The flowers tend to have short, curved nectar spurs. And the bee pollination syndrome is ancestral. So the first columbines to migrate to North America from Asia were bee pollinated. But then there were 2 independent evolutionary shifts to hummingbird pollination syndrome. So that means that red pigment, straight nectar spurs to accommodate a hummingbird beak, and exerted reproductive organs, they all evolved twice separately in this genus, which is so cool. And then from there were 5 independent transitions to hawk moth pollination. So 5 times in the genus, there was convergent evolution of even longer spurs, a shift in flower orientation, loss of floral pigment to help with detection at dusk when hawk moths are active. And all of this happened in less than 3 million years, which I know sounds like a super long time, but it's very fast evolutionarily speaking. So up until this point, our lab's work has really been focused on species that make straight spurs. We know next to nothing about the curved spurs of bee-pollinated species and how they developed. And in order to understand the rapid radiation of this genus, we need to study that curved ancestral nectar spur to find out how it shifted so many times to produce the diversity in spur morphology we see today. So that's the focus of my dissertation research. What are the developmental and genetic bases of curved spurs? And how did those things evolve during the shift to hummingbird pollination when the spurs needed to straighten out? To answer these questions, I'm working primarily in this pair of sister species, bee-pollinated Brevistila and hummingbird-pollinated Canadensis. Sister species means they're more closely related to each other than to any other species of columbine. And they also represent one of the two independent transitions between pollination syndromes. I'm going to share a few stories about them today that represent the different chapters of my dissertation, but they're all very much interconnected. So let's start with chapter 1, where I characterize the genetic architecture of floral traits in these species. So how many genes control a trait? Where are they in the genome? And how much of an effect do they have? So many floral traits shift during pollination syndrome transitions. And while the focus of my thesis is spur morphology, spur shape, length, it's important to understand how it's evolving in concert with all of these other traits. So to do this, we use a method called quantitative trait locus mapping, which I know sounds like a big mouthful, but a quantitative trait is just a trait that can be measured and that varies along a continuum. So like flower color or nectar spur shape. A locus is just a location in the genome, and mapping means we want to find out where it is. So QTL mapping experiments start with a cross of the species we're studying, which we call the parents, to make a hybrid plant, which we call an F1. We then cross the F1 to itself to make an F2 generation. And our F2 generation consisted of 350 plants and was absolutely stunning. So please admire them while I have a drink of water. But we need some things from these F2s. We need a lot of data. So we need genotype data, which is DNA sequences, and then phenotype data or trait measurements. To get our genotype data, we sequenced the genomes of all the F2s as well as the parents. So this is Angie Ballerini, who started this cross and mentored me through this whole project, and my lab mate Minya, who's also used this data set for her thesis. And we're doing hundreds of DNA extractions from our plants and getting them ready for genome sequencing. And then we get the data back. So I'm going to represent the parent genomes as these like abstract pairs of chromosomes, though in real life, Aquilegia has 7 pairs. Their genomes have been recombined in the F2s. So by comparing F2 and parent genotype sequences, we can identify which chunks of the F2 genome inherited both copies from Canadensis, which chunks of the F2 genome inherited both copies from Brevistila, and then which chunks inherited a copy each. So at the end of all this, we have 350 F2 genome sequences with all of their chunks assigned to one of the parents. So that's our genotype data. Now we need our phenotype data. For this chapter, I measured 17 floral traits in our F2 population that span floral color, nectar composition, and organ shape and size. I dissected the F2 flowers and scanned them, and then used MATLAB and ImageJ to measure color and organ size from the scans. And then collected nectar with capillary tubes to measure its volume and concentration. For each trait, I took an average across 3 flowers per F2, but most flowers have 5 of each organ that all need to be measured. So if we do the math, it's about 90,000 trait measurements. So it was an insane year or two of data collection. And I just want to take a moment to thank Joanna and Angie, who were 2 amazing undergraduate students who helped with all of this data collection. I'm only going to be able to focus on spur length and curvature today, so I do encourage you to check out our publication in Evolution if you want to know more about the other traits, because there are some really cool stories in there that I'm very sad I don't have time to share today. So let's start with spur length. I have my 2 data types ready to go, and now it's time to generate the QTL map. So I'll go through a simplified example of what this looks like because it's a really cool method, and I think it's nice to know what's going on. So here are 4 F2s. 2 with long spurs like Canadensis, and 2 with short spurs like Brevistila. And here are their genomes. So this mapping analysis is going to scan the genome for correlations between genotype and phenotype. So let's start at this first location. Here we see that the long-spurred F2 has the long parent genotype, but the other long F2 has the short parent genotype. While this short F2 has inherited one copy from each parent, and the other short F2 has the long parent genotype. So we would consider this location to have a low correlation between phenotype and genotype. It's all good. Like, let's try another one. So we have long F2 short parent, long F2 both parents, short F2 long parent, short F2 short parent. Again, low correlation. That's okay, we're gonna try again. So we have long F2, long parent, long F2, long parent, short F2, short parent, short F2, short parent, woohoo! So at this location, there is high correlation between phenotype and genotype. Short F2s consistently inherit their genes from short Brevistila, and long F2s consistently inherit from long Canadensis. So this locus, this location, contains the gene or genes responsible for spur length. And then a little later on, we can see correlation is low again. So if we were to plot the strength of the correlation between spur length and genotype across this chromosome, it would go like this. Low, low, high, low. And if we connect the dots, you can see a little mountain where our correlation is high, and that's a QTL map. So instead of X marks the spot, the mountain marks the spot. But Achelicia does have 7 chromosomes consisting of 300 million DNA base pairs, and we used 350 F2s instead of 4. So making the real QTL map requires a lot of computing power, and it looks like this. So along the x-axis are the 7 chromosomes in a line, and along the y-axis is the strength of the correlation between phenotype and genotype. And you can see there's a little mountain on 6 of the 7 chromosomes, indicating that at least 6 loci are responsible for controlling spur length. So this trait has what we would call a very complex genetic architecture. Lots of genes are contributing. The size of the mountain corresponds to the size of the effect the locus has on a trait. So this locus on chromosome 7 is responsible for 16% of the variance in spur length in the F2s, while this much smaller mountain is only responsible for 3%. Okay, now we're going to try this with spur curvature and see how the maps compare. But first, we actually had to figure out how to measure spur curvature, and it turned out to be so complicated that we needed to collaborate with some applied mathematicians. So this is Gary Choi. He was a student in the Mahadevan lab, and he developed our spur curvature quantification pipeline. So here's how it works. We start off by identifying a straight spurred Canadensis petal to serve as the reference for all of the measurements for our F2 petals. Then we take the F2 petal we want to measure, and we normalize both spurs for length so that the attachment point becomes 0 and the nectary becomes 1. The pipeline then measures the slope of the reference and F2 spurs continuously between these 2 points and plots them to get 2 curves. And I just want to point out that we're actually only taking the measurement from the bottom half of the spur, because there's a lot of stuff going on up here with spur shape that's not really relevant to curvature. And so we only wanted to capture the bit that actually shows curvature. So that's why this plot is starting at 0.5 to represent the bottom half of the spur. Okay, so then the pipeline calculates the difference between these 2 curves. And then that difference value becomes the phenotypic value we'll use in our QTL mapping. So a straight F2 has a low curvature difference value because it's similar to the Canadensis reference. And then a really curvy F2 petal will have a high difference value. Okay, so now we have our phenotype data. We're ready to do our QTL map. And here's the result. Okay, this is a very different map from spur length. There's really just a single giant locus responsible for spur curvature. It explains 46.5% of the trait variance. So the other 50% is likely from, a couple of these really tiny loci, plus ones that were even too small for the analysis to pick up on at all. But it still presents the very exciting possibility that we could find the gene that causes curvature. So you might be wondering, why don't I know what that gene is right away from this QTL map? And that's because the peaks are really only giving us a very general location on the chromosome. So each peak can span hundreds of genes, and only one or a small handful might be the actual gene that we're looking for. So we're going to come back to this later on, so stay tuned. But just to put this result in a wider context, finding morphological traits that are caused by single large effect loci remain rare in the QTL mapping literature. So this was a really exciting result. Very rare. Additionally, the curvature peak doesn't overlap with the spur length peak on chromosome 7, but they're pretty close together. And there's also a large QTL for pistil length right next to them. They're all within a few centimorgans of each other. So these are 3 morphological traits that are extremely important for pollinator interaction and reward. And their linked QTL could possibly have contributed to the rapid evolution of pollination syndromes because they're likely to be inherited together. Linkage of major QTL is common throughout the pollination syndrome literature too. We see it in Petunia, in Penstemon, in Mimulus, suggesting this could be a shared mechanism for rapid pollinator-driven radiations. So in chapter 1, we've gotten a broad view of the genetic architecture of spur length and curvature and how their relationship to all these other floral traits. But now in chapter 2, I'm drilling down to understand the developmental and transcriptional dynamics of these different spur morphologies. So let's start with development. Here's a Brevistila petal, and throughout this discussion of spur development, I'm going to be referring to this surface as proximal and this surface as distal. And in order to generate a curved spur, there needs to be differential growth between proximal and distal surfaces, right? The distal surface needs to be longer so it can curve around. One of my main questions was, what's the nature of this differential growth? Is it achieved through differential cell division, elongation, or both? How did that growth pattern change in order for straight spurs to evolve during the shift to hummingbird pollination? And finally, are these patterns conserved across species and across independent pollinator transitions? So to answer these questions, I counted and measured the length of epidermal cells in a single file along the entire proximal and distal surfaces of Brevistila and Canadensis petals. And this is what I found. First, Brevistila spurs have 24% more cells in their distal surface than proximal, but there's no significant difference in cell length between the surfaces, indicating that differential cell division could be the driving force behind curvature. So I thought, okay, maybe straight spurs evolved by equalizing cell division between the surfaces, but no, Canadensis petals still have way more cells on their distal surface than proximal, and instead, their proximal cells are 19% longer than their distal cells to compensate and straighten out. So that's what's going on in this species pair representing this one transition from bee to hummingbird pollination. But what's going on with the other one? Do these patterns appear convergently in the second transition? So to find out, I counted and measured more cells in Saxomontana, which is another curved bee pollinated species, and in Formosa, which represents the second transition to hummingbird. And indeed, Saxomontana is curved due to differential cell division, and Formosa straightened out by elongating its proximal surface cells. So it converged on the same mechanism as Canadensis. One surprise, however, was that the overall spur length difference between species is associated with cell number. So all of these species are around twice as long as Brevistila and have twice as many cells, but cell length across the 4 species is comparable. And this was really surprising because previous work from our lab has indicated that cell anisotropy was responsible for spur length diversity in Aquilegia. But that study did focus on moth-pollinated species that have much longer spurs. So it could be that spur length diversity in bee and hummingbird species maybe happens via cell number, and then the transition to hawk moth pollination involved cell elongation. It could also just be unique to these species that I selected for this study. Brevistila is by far the shortest spur we've ever looked at. But on the whole, this was turning out to be a way more complicated and interesting story than I originally thought it would be. And we were only just getting started because next I wanted to find out how the petals got to this point. So I did more cell counting and measuring at earlier developmental stages in Canadensis and Brevistila. So for all the data I'm about to show you, developmental stage is represented as % of mature spur length. So let's start with Brevistila cell number. What really puts distal cell count ahead is the duration of cell division. So the proximal surface reaches its maximum cell number when the spur is only 30% long, whereas the distal surface doesn't level off until 50%, and you get that big gap. If we compare that to Canadensis, the data are a little messier, but it appears that the surfaces are leveling off at roughly the same time, unlike Brevistila, and maybe the steeper slope in the distal surface suggests a higher rate of cell division. So while we found that differential cell division is conserved between curved and straight spurred species, these results make us wonder if the mechanism by which it happens may have changed during the evolution of hummingbird pollination. So looking at cell length next, in Brevistila, cell elongation just proceeds at a steady and equal pace between the two surfaces. But in Canadensis, we start to see differential cell elongation between the two surfaces at around the 30% stage, so that by maturity, there are much longer cells in the proximal surface than distal. If we combine the plots for the two species, we can see just how dramatic that overall cell number difference is. And then it's actually already established by the earliest stage at which we sampled. And I want to particularly bring your attention to the 10% stage in Canadensis and the 20% Brevistila stage. Because at these stages, their spurs are overall the same length, around 2 millimeters, but Canadensis spurs have way more cells than Brevistila. So this really raises some fundamental questions about cell recruitment to the nascent spur and cell division rate at extremely early stages. So we want to look into this more. In contrast, cell lengths are quite comparable between species, and it's really this subtle shift that results in the differential elongation in Canadensis. So now that we understand the developmental dynamics of spur shape, I want to see how they play out transcriptionally, which means what's going on with the RNA. So we spent a lot of time in chapter 1 talking about genome sequences, but that's DNA. All cells have the same genome, the same complete set of columbine genes, but not all cells are expressing the same genes. So expressed genes are transcribed from DNA into RNA. So then we sequence the RNA, also called RNA-seq. We can find out which genes are being expressed, at what level they're being expressed, and hope this can give us some insight into the molecular basis of the developmental phenomena that we just characterized. So for this RNA-seq project, I sampled both species at 4 developmental stages, again, based on % of mature spur length. So here are the petals at a slightly larger scale, so I can show you how I did the tissue sampling. I first dissected away the blade and then separated the proximal and distal compartments of the spur. Each biological replicate consisted of all 5 proximal or distal spur halves from the same flower. I collected 5 replicates each, so there were 80 samples total, 2 species, 4 stages, 2 tissue types, proximal, distal, and 5 reps. And then we sequenced their RNA. And for a first look at the data, I'm going to start all the way zoomed out and show you a principal component analysis, which is just an unbiased way to assess how all of our samples are clustering. And this is looking pretty good. So PC1 is separating out our samples by stage, and then PC2 by species and a little bit by proximal distal. And I decided to remove a couple of reps here and there that were coming out with slightly the wrong stage, but overall, the stages are clustering nicely with each other and they line up well between species. So this tells me I didn't mess up all of those dissections, which is a big relief. But with a sampling stage as complex as this, there are like seemingly infinite ways to analyze and explore the data. Here are some analyses that I did if you're curious, but don't worry, we're not going to go through them in detail today. I don't want to show you endless heat maps and gene lists. I just want to share some key takeaways and themes that are relevant to our main story today. Namely, there was loud and clear developmental signal in this RNA sequencing data that reflects our findings from the first part of this chapter and gives us insight into the molecular processes underlying them. So overall, we found that the proximal and distal compartments of the spur are experiencing very different gene expression environments, with several thousand genes showing different expression levels between them. There was some indication that cytokinin signaling could be involved in the differential cell division that we see in both species. Cytokinin is a hormone that's a known promoter of cell division. And we saw lots of transcriptional evidence for cell elongation, cell wall synthesis in canadensis proximal tissue, and repeated indications that auxin signaling, which is a known promoter of cell elongation, could be playing a role. Okay, we're at the brink of chapter 3, where we're going to synthesize the data sets from the first 2 chapters to try to find the gene that causes spur curvature. So from chapter 2, we learned that differential cell elongation is the critical developmental difference between curved and straight spurs, and that the hormone auxin could be playing a role. And actually, our lab has already done some work with auxin and spurs. So these flowers have been through an experiment that turns off auxin response genes. The experiment also turns a pigment gene off as like a visual marker. So when we see white petals, we know their auxin genes are off as well. So the white petals here have shorter spurs due to shorter cells, meaning that auxin promotes cell elongation in the spur. We also know that brassinosteroids, which are another type of plant hormone, regulate cell elongation in the spur as well. So applying brassinosteroid causes longer cells and longer spurs, and turning off brassinosteroid response genes causes shortening and distortion of the spurs. So with all that in mind, let's revisit our QTL map for spur curvature. So this peak on chromosome 7 contains 199 genes, which is a lot of genes. So a great way to narrow this down is to ask how many of these genes are expressed in our RNA-seq data set, because presumably the gene that is responsible for spur curvature is expressed in the spurs. Okay, so if we do that, it turns out that 144 of the 199 genes are expressed. So that narrows the list by a decent amount. It's still a lot of genes, but not so many that I couldn't go through them all and look at their known functions in other systems to choose a promising candidate. So that's what I did. And there was really one gene out of the 144 that stood out from the pack. And that is IBL1, or increased leaf inclination binding BHLH1-like. Yikes. But it is a transcription factor, which is a gene that regulates other genes. It's known to repress cell elongation in Arabidopsis, and it acts via the brassinosteroid and auxin pathways. Whoa, so this was really exciting. And knowing this about its function, I got even more excited when I saw IBL1's expression in my own RNA-seq data set. So on the x-axis are the developmental stages that I sampled, and on the y-axis is the gene expression level. So overall, IBL1 is more highly expressed in Brevistila than Canadensis. And then within Brevistila, it's more highly expressed in the proximal compartment of the spur. So this suggests a model in which IBL1 represses cell elongation in the proximal compartment of curved Brevistila spurs. But reduction of expression in Canadensis relieves the oppression and allows promotion of cell elongation in the proximal cells via brassinosteroid and auxin signaling, and thereby straightening the spur. So that sounds great, but like, how do we test that model? So first, let's just take a look at IBL1 expression in Brevistila flower buds. This is another in situ hybridization, like we saw at the beginning of the talk, but this time where you see purple, it's IBL1 that's being expressed. So in this very early flower bud, we can see IBL1 expression throughout the entire petal. But at this later stage, which represents something in between the 10 and 20% stages from my RNA-seq data, you can see that expression is more specific to the spur, and that it even appears higher in the proximal compartment. So this is a nice confirmation of my RNA-seq data. But then to test IBL1's function, we have to turn it off in Brevistila and see if its spurs become straighter. To do this, we use a method, we use a method called virus-induced gene silencing, and it only works after you've grown your plants up to the right developmental stage. So the plants take about 9 months to grow. I had carefully planned all of my fall 2019 plantings, so I'd have tons of Brevistila ready for my experiments the following spring and summer, and then COVID happened. So I had to throw away all of my plants, start again from seed once we were allowed back in the lab. And around 1 year later, I was finally able to do our VIGS experiments to silence or turn off IBL1. Okay, so first, I'm just going to show you a flower that has been through no VIGs, just as a reference. As I mentioned before, when we do VIGs, we also silence a floral pigment gene so that white petals are this visual cue to us that our experiment worked. So next, I'm going to show you some control VIGs plants that have only had their ANS pigment gene shut off, not IBL1. And you can see that there are sort of varying levels of silencing. So in this flower, sorry, oh no, I spoiled it. Okay, in this flower, we can see that all of the petals are white. So that's an example of strong silencing. But then in this flower, which is these petals from a different flower, have patchy silencing. So only like 2 of the 5 petals are white. And that's okay. Patchy silencing is very, very common in VIGs. But the main take home here is that these petals have the same shape and size as the petals that have not been through any VIGs. Because now I'm going to show you plants that have been silenced for IBL1 as well. Again, we see varying levels of silencing from patchy to strong, but the silenced petals appear a lot straighter than their non-silenced counterparts and the control flowers. So this was a really exciting result. I was super jazzed when I saw these straight spurs. But I will say, there is some variation in how curvy Brevistilla spurs are, even when they haven't been through VIGs. And our gene is predicted to only be responsible for like 46% of spur curvature. So I wanted to repeat the VIGs experiment in a species with bigger, curvier petals in case the phenotype would be more obvious there. So I chose to do that in Aquilegia vulgaris, which is a bee-pollinated European species. But the seeds turned out to be some kind of weird horticultural mix because they wound up being a lot of different colors, including pale pink. So it was a little harder to see our pigment silencing marker, but that's okay. So for COVID reasons, again, I was not able to perform the ANS only pigment controls. So you're just seeing IBL1 VIGs here. But again, varying silencing, but we're seeing results pretty consistent with what we saw in Brevistilla. The silenced spurs are straighter than their non-silenced brethren. So we were really excited about this. These were extremely exciting results, but unfortunately we can't just get away with saying like, hey, the spurs look straighter, so I totally discovered the gene. We have to do a lot of quantification. So we have to quantify the level of silencing. Did we actually turn IBL1 off in these petals? We have to quantify the phenotype. So are silenced petals actually straighter than control petals, or is this just natural variation in Brevistilla? And did they straighten out in the way we expected based on IBL1 function? Are their proximal surface cells longer than their distal ones? But all of this quantification requires a ton of silenced petal tissue because you need multiple replicates of everything to be able to do statistics on the data. Doing VIGs in these species, though, is much trickier than in the small horticultural hybrid we usually do our VIGs experiments with. The transformation rate is really low. It's like 6%. So even though I spent months doing VIGs on hundreds of plants, in the end, I only had a few that showed any silencing. And there wasn't enough for any of the quantification I wanted to do. So I was able to perform a qualitative assessment of IBL1 silencing in Brevistilla using a method called RT-PCR. So what does that look like? Well, we have 8 samples here that are repeated between these 2 panels, 4 from IBL1 silenced plants, and 4 from our control petals that only had the ANS pigment gene turned off. And we want to compare IBL1 expression between them. But first we have to look at a control. We looked at expression of IDI, which is this housekeeping gene. It's just super highly expressed all the time, always in all the tissue. And the darkness of the bands here indicates the strength of expression. So they are all the same shade across those 8 samples. And that just tells me that I used successfully the same amount of petal cDNA across the 8 samples. So now that I know my controls look good, we can then look at the levels of IBL1 expression in our samples. And we can see that the bands in the control tissue are darker than the bands in the IBL1 VIGs tissue. That means there's less expression in this tissue, indicating that I did, in fact, successfully silence IBL1. So I believe those straight spurs a little bit more from my VIGs plants now that I see this evidence for successful silencing. So this is a really promising start. We're really excited about IVL1, but I will admit it's a little heartbreaking that I wasn't able to confirm for sure that it is the curvature gene. But reflecting back on it, I'm not sure I would have been able to, even if I hadn't lost a year to COVID, because VIGS just isn't the right tool for the job here. It doesn't generate the tissue we need to do the quantitative assessment of the silenced petals. But luckily, Elena just got a grant to develop stable transgenics in Achilles, so we're hopeful we'll have better tools very soon. So I've learned a lot of things from the Columbine flowers over the last 6 years. How their many traits shift together during pollination syndrome evolution, the emerging complexity of their developmental and transcriptional dynamics, and the potential simplicity of a single gene that controls spur shape. But what I want to end with today is, what more will we learn from the flowers? I'm really proud that my dissertation work will lead to so many new projects. The chapter 1 QTL mapping study alone is going to keep everyone busy for many years to come. So we identified a ton of candidate genes for flower color and nectar composition that need to be validated. We still need to determine the developmental basis for many organ size and shape traits. We've only gotten spur length and curvature done so far. For chapter 2, I was using a 2D imaging technique to measure cells in a 3D organ. So how do cell number and shape change around the circumference of the spur? And what's happening in even earlier developmental stages when species cell number differences are established? Our RNA-seq data also indicated that vasculature could be playing a role in spur development. I had focused on epidermal cells. So this is a super intriguing avenue for future exploration that we're excited about. For chapter 3, we obviously want to confirm whether IVL1 is the curvature gene, but it's also implicated in a complex regulatory network with many closely related genes. So we want to explore this network further to see if its other players are involved in spur shape in this system, maybe in other species, independent transitions between pollination syndromes. It's going to be really cool to pick this apart some more. So I can't wait to see what things we learn from Aquilegia flowers next that will help us understand how and why there are so many different, complex, beautiful flowers on this planet. There are so many people, experiences, and organizations that made it possible for me to spend the last 6+ years with my flowers, like my department and my professional society and my funding sources. But before I get ahead of myself, I do want to rewind and talk about some earlier parts of my life that helped me get here and to become a better scientist and human. So first, I want to thank Putnam County 4-H because it changed my life. I learned video production and public speaking and puppeteering from 4-H, but most importantly, I experienced firsthand the power of university outreach programs and what happens when educational institutions build service and extension into the foundations of their mission. And I'll carry these with me always, as well as the impact of the mentorship that I received from Dina and the other 4-H leaders. I'd also not be where I am today without my high school. It was such a special place for me. I was awkward and I was shy and figuring myself out. And my teachers guided me through it with such grace and kindness, especially Doc and Mary, who became my family. Dr. Dyack, my incredible advisor, and Miss Merrill, who's the reason I became a plant biologist. So thank you. I'm grateful for the scientific mentors I had the privilege of learning from prior to graduate school. And Mike was randomly assigned to be my advisor the day I got to Cornell, and little did I know he would introduce me to the field in which I'd eventually do my dissertation research. And the Scanlon Lab is where I met Margaret. She mentored me through so many firsts. First RNA extraction, first project, and I'm so grateful for our continued collaboration and friendship. Greg and Rob at NYPG guided me through my first science jobs. They introduced me to the magic of the garden, and I benefited so much from their kindness and support. And in the Cruzy lab at NYU, my skills and knowledge just grew exponentially, and I'm eternally thankful for how well Gloria and her amazing lab prepared me for grad school. NYU is also where Science IRL began. I had no idea how much it would ultimately shape my life and career, and I'm so lucky that Silviana and Ramin took the plunge and went on this wild adventure with me, and that Milo and Matt joined the team once I got to Harvard. It's been an honor and a privilege, you guys. And many thanks to the dozens of guest scientists who have appeared on Science IRL over the last 6 years, including almost all of my committee members. Scott is excused because he's in California, but Scott, I owe you a Science IRL episode. And thank you to our funding sources for keeping us afloat and able to do what we love. So graduate school is the longest I've ever spent in one place. And what I loved most about it was its many built-in mechanisms for finding community. So thank you to the Radcliffe Choral Society, the Action Team, and Lab Exchange for helping me find my voice in very different but powerful ways. I'm so grateful for the community of plant biologists in our department, especially at the Arboretum. The ARB literally saved all of my plants for the QTL mapping project, and I cherished the time I got to spend there with all my favorite plant people. And I don't know how to thank my beloved cohort. Gosh, even at our blizzardy interview weekend, I think we knew this was going to be something really special. You're just the best. I'm so grateful for all of you, and I'm still heartbroken that COVID robbed us of our last year together. But I know we'll be apples and nuts forever and can change at any time. Thank you to my incredible committee, Robin Hopkins, Scott Hodges, and Ned Friedman. I've always come away from our meetings invigorated and ready to tackle the next set of experiments and analyses. And you've each helped me immensely in moments of need and have made me a better science storyteller. The community that most shaped my day-to-day life in grad school is, of course, the Kramer Lab. You guys have just been the best to me, and I'm an only child, but I'm coming away from the Kramer Lab with so many sisters. Angie, I still remember awkwardly coming into the tea room to ask Angie about projects my first year, and that conversation turned into years of collaboration, friendship, and growth. I'm so grateful for Angie and all she's done to help me and my PhD reach its full potential. Claire made me feel so welcome when I was a nervous G1 by setting the tone for the lab and making it feel like we were part of something cohesive and special. And it was so inspiring to see her shine through all the major grad school milestones and to have an example to look up to. And what a lucky day for us when Steph joined the lab. I am amazed always by her skills. I'm a better scientist because of Steph. But most importantly, I couldn't have asked for a better friend during the pandemic and the last months of wrapping up our projects. And what can I say that will fully communicate how thankful I am for my lab twin, Minya? This is a bond that only we share. It's magical. And I would never have gotten through grad school without her. I think one of the coolest jokes that academia plays on you is that it brings people into your life who become your family as you share these like super intense experiences together. And then after a few years, you have to leave. And I was dreading this, like, what's going to happen when me and Minya graduate? And I was so relieved that when she took a postdoc in Connecticut, because I can always visit her whenever I need some lab twin time. And it seems like an impossible task to properly thank Elena. I can't imagine doing my PhD with anyone else. I just grew in leaps and bounds as a researcher under Elena's mentorship and was constantly in awe of her scientific mind. And through her example, I learned about mentorship and how to help people see the joys of plant biology. And I left every meeting with a sense of clarity and feeling supported and understood. And Elena was always there for the lab, even while navigating OEB through multiple crises, including the pandemic. So she really is Science Wonder Woman. All of these adventures have brought so many friends into my life. I'm full to the brim with love and appreciation for you all. And I want to especially thank Kara, Jess, Kaya, and Crystal for making Somerville home for the last 6 years. And I could write a whole extra dissertation on how thankful I am for my family and how much they've carried me through grad school. Especially my Nana, my grandma, and my Aunt Bee, who are the matriarchs of my family. And I'm so proud to come from such wise, strong women. And thanks to my mom and dad. I don't know how I got so lucky that the universe put us all together. All I am is because of you, and I love you infinity and beyond. And now that I'm a little verklempt. It's a great time to answer your most rigorous science questions. Thank you.
Now we'll give Molly a moment to gather herself. We can all clap for Molly. How much we love Molly. And your mascara is holding in just fine. So
Yes,
don't,
it is.
worry, don't worry about it. You're totally good. She's very concerned. But we would love to take some questions.